So the next topic would be biosensors in post-harvest management from Dr. Vivek Kamampati, Assistant Professor in the Department of Food Process Engineering, NIT Rourkela. Right. So these are the contents for the presentation. So I will be introducing little bit about post-harvest management, and then biosensors. What are biosensors? And then how do we use these biosensors in the post-harvest management, and what are the challenges? Different types of biosensors and how to use these biosensors, and where are the, what are the applications of these biosensors also we will try to get. And then in the final trends and future implications we will try to learn, right? So the in the morning class you must have learnt about what is post harvest management, what is post harvest engineering, what are the post harvest practices, right? So in a nutshell, if I tell what is post harvest management, it is the processes and practices applied to agriculture produce, right? Any kind of agriculture produce, let it be fruits, vegetables, grains, whatever the agriculture produce you take, there is a set of process and practices produced after it is harvested to maintain the quality, extension of the shelf life, reduce losses and ensure the food quality, right? So this post harvest management, it includes whatever the steps involved in the supply chain, starting from the after harvesting we will transport or we will keep in some places, okay, temporary storage, permanent storage, like handling, storage, processing, packaging, transportation and marketing of fruits and each and every link in the supply chain will be considered as the post harvest management, right? So if you see what are the key aspects of post harvest management, so definitely first one is handling. Why handling is a key aspect? Any idea? In the post harvesting, the moment you take out from the field, right, you need to store. Say, take an example of any fruits or vegetables you will harvest. After harvesting, immediately what we will do, we will go for the pre-cooling treatment. So why pre-cooling is required? To reduce the respiration rate, right? So after that, what you are planning? So are you sending the products directly to the market or through intermediary, or how you are handling this particular commodity? That is important, right? So that is all about handling. The second one is sorting and grading. What do you mean by sorting and grading? So after harvesting, definitely these agriculture commodities, they are they are not uniform in size, shape, right? So what we will do, according to the size, shape, or maybe the economic value, okay, take an example of egg. The egg size is different, right? Small, medium and big size, okay. When we say sorting, based on the physical characteristics, sorting will happen, and grading means based on the money, based on the economy, right? Based on the value of the product, grading will happen, okay. That we have to ensure. The third one is storage. Definitely storage is very important, especially in terms of grains, right? After harvesting, take an example of paddy, will harvest and then we'll reduce the moisture content below 20%, and then we'll go for the storage, right? The last one is transportation, okay, where you are transporting, okay. Something like FCI godowns will be there, and different big storage vessels, or maybe companies who are handling the storage technologies, we have to send these agriculture produce to that place, right? So these are some of the key aspects. So what are the challenges? In each and every aspect we have a lot of challenges, okay, especially that's why in India these post harvest losses are very huge. As a technocrat, as a food technologist and agriculture engineering people, we have to understand what are the different challenges associated with post harvest management, right? Otherwise we cannot control those losses, okay. The first one is handling loss. Definitely mechanical damage will be there, lack of skilled labor, inadequate tools and equipment, infrastructure facilities, that is associated with handling. The second one is sorting and grading, right? Sorting and grading, inconsistent standards. Standards are very much important, okay. Then labor intensive process and technology access, and then storage. In the storage, especially relative humidity and temperature is very much important. Take an example of grain storage, right? if you store grains in bulk, definitely the heat will be generated inside the grains. Okay, during summer season, what is the criteria for storage? During winter season, how we need to store? Depending on the seasons, depending on the harvesting time, okay, depending on the material, agriculture commodity, we have to decide what type of storage is required. That's why temperature fluctuations are important. The very big issue is with pest, rodent, fungal infestations and lack of cold storage infrastructure. Okay, lack of cold storage infrastructure, I will give you one example. Lot of exotic fruits are available in the northeastern part of our country. Say suppose for example Arunachal Pradesh, Assam, or maybe Tripura, sister states, northeastern parts, but we are unable to bring those fruits to even south or maybe in the central part of India because of lack of cold chain infrastructure. That's why government of India, they are pushing hard for developing the cold chain infrastructure, right? That is one major criteria that we need to focus. The third one, of course, fourth one is packaging. Why packaging is very much important? Packaging, the functions of packaging are very much important. If you are not packing any food, so it will not be attractive to the customers, right? Customers will like when the product in the packaging material is looking good, maybe attractive, labeling, and then whatever the nutritional information. As a customer, I feel like reading what are the nutritional information available on the packaging. That is also very much important, right? Then cost constraint, and then the environmental impact. Definitely, because of the environmental impact, a lot of researchers, they are coming up with biodegradable packaging and lot of alternative packaging solutions, right? The next one is transportation. Transportation is also very important. I will give you one more example related to these tomatoes, right? During winter season, the prices of tomato will come down normally. It did not definitely meet the demand supply gap. Why that is happening? Demand supply, why there is a gap, huge gap between demand and supply? I'm asking you people, any idea in terms of tomato? Why I'm telling this? If you take Kolar region in the Karnataka, one of the biggest producers of tomato. Every year they dump tomatoes on the road. Okay, huge losses incurred, from these losses, especially for the farmers. They invest lot of money for cultivating these tomatoes. Okay, and then they get the crop but unable to sell it in the market. The second biggest market for the tomato in our country is in Chittoor. Kolar and Chittoor, two are biggest markets for tomato. If you go there, you will understand the scenario. Lot of losses are incurred because of the losses of this tomato. Why? Because lack of transportation available. The trucks are not available. Maybe because of rise in petrol, diesel prices, it will affect the price of the commodity. Okay, farmer, as a farmer, they have some expectation, but in the market, because of huge supply, the prices will come down. Okay, these are some of the issues that we need to consider as a food technologist or agriculture engineer peoples, right? So how to solve these problems? By doing all these things, right? The first one is training and capacity building. Definitely we need to train the man force how to handle the post harvest losses. The second one is improved storage facility. Storage facility has to be improved, and the infrastructure, storage infrastructure also very much important, right? Storage infrastructure, something like cold storage, right? So government is providing subsidy for that. If somebody is interested to establish cold storage firms, okay, that is important. Then smart packaging solutions, definitely packaging is one criteria. And then the last one is adopting the biosensors. Okay, what do you mean by biosensors? We'll learn. The use of biosensor to monitor ripeness, detect spoilage, pathogens, environmental conditions. Okay, environmental conditions like temperature, humidity, and ethylene levels in real time to ensure the quality and reduce the losses. Okay, so in this lecture we are going to focus only on the adoption of biosensors. Okay, though post harvest is very huge topic, and there are a lot of issues with the post harvest management. Lot of challenges are there. There are so many solutions. Okay, there are so many ways to solve the problem, but via biosensors, how we are going to solve the issue, that we will be learning in this particular lecture. Okay, so what do you mean by biosensor? Biosensor is nothing but it's a device. Okay, analytical device. It combines biological component with a physicochemical detector to measure a specific analyte. Okay, say suppose for example when I say analyte or substrate, okay, all of you must have seen this glucose sensor, right? So at home, those who are, your parents or maybe elder people in your home are they are diabetic, so they measure glucose at regular intervals. So what is the principle of that particular glucose? They take the blood sample, drop of blood will be dropped on the sensor. Okay, so via that sensor electrochemical process, okay, it will trigger, it will trigger some of the electrons, protons will be released, hydrogen peroxide will be released. How much hydrogen peroxide has been generated? Okay, so based on that concentration, the numerical value will be generated. Okay, so here the glucose is analyte, okay, or substrate. Using the sensor, you are identifying what is the quantity, what is the concentration. Okay, ensure safety and quality of the food product. Definitely, food quality monitoring plays a crucial role in identifying and mitigating the risk. Okay, and then contaminants, adulterants, and other factors that can compromise the integrity of the food supply chain can be controlled by using this biosensors. Okay, so how it got emerged? Traditional, what is the traditional practices? Usually in the laboratory, right, in the food safety quality lab, you take the random sampling and destroy the sample. You analyze, what is the physicochemical properties of that particular, product. Okay, you cannot tell accurately in a lot of 100, 100 kgs or maybe 1000 kgs. If you take industry, go to industry, they will be handling in a bulk, right? So each and every sample you cannot tell what is the quality of this particular sample. Okay, if you use these biosensors, you can attach some corner and according to the maybe respiration, production of the ethylene gas, or maybe through any gas, because what will happen, fruits and vegetables, they respire after, even after harvesting, they produce some gases. If you measure the gas, what type of gas is being generated from that, accordingly using the biosensor you can identify easily. Okay, whether what is the ripeness of that particular product, or how long we can store in that particular relative humidity and temperature, those things we can easily. So these biosensors directly or indirectly help us to take the decision. Okay, how long to store, where to store, when to transport, using what mode we have to transport. So many decisions we can take using these biosensors. Definitely, in the traditional practice require time. Okay, if you want to find out how much glucose is there in the sample, how do you do analysis, right? You will extract the sample, you will prepare the sample, and then using some instruments you will measure. Okay, if you are doing HPLC, okay, it will take 2 hours, 3 hours time. Definitely time consuming process. Using biosensors you can minimize the time for determining all these things. Then delay the detection of hazards and increase the potential of consumer exposure to harmful substances. Right, nowadays there is a big issue with pesticide residues. Lot of spices, we are one of the major producers of spices, right? So lot of spices have this problem, pesticide residues are more. Okay, because of that reason, lot of spices are being rejected by the European Union. Okay, that's why we need to establish a good technology which can detect the pesticide residues without destroying the sample. Biosensors can do that work. Okay, so analytical device, these analytical devices integrate biological recognition elements with transducer components, helps in detecting and quantifying specific analytes in the food samples. Okay, we'll learn what are those. Okay, then what are the importance of biosensors? Definitely it is important for maintaining the integrity of the food sample, food safety and quality assurance. Okay, then nutritional monitoring, definitely we can monitor the nutrition, how with respect to time, how the starch is converting as a simple sugar, or maybe how different things are converting. Okay, with respect to time we can easily tell. Spoilage and freshness detection. Okay, some volatile components, volatile organic components will be released to monitor the freshness of the perishable foods, that we can easily identify using this biosensors, right? Next one is process optimization. So Process optimization, suppose for example, if you are doing any fermentation process, lactic acid bacteria or maybe any kind of fermentation you are doing in the lab, okay. So what are the criterias, okay, what are the different criterias, temperature and then relative humidity required for that particular fermentation process, okay, how long we need to keep, all these decisions we can easily take by using different parameters. Biosensors, it will help us in optimizing. Suppose for this particular fermentation process we need 22 hours or maybe 26 hours, okay. So based on the previous experiment, we can generate results and we can monitor and control critical parameters, okay, enzyme activity, fermentation progress, and then pH changes and all we can easily edit. Shelf life prediction, definitely shelf life prediction is very much important. So why? Because in an agriculture, I told you one example, right? Tomato, perishable products, tomato after harvest it will be ripening very fast, okay. So almost for 2 weeks you can keep under the ambient temperature, okay. Before that you need to decide whether you will be selling the product which place, okay, 100 kilometers from the cultivation area or maybe 1000 kilometers or maybe 2000 kilometers. 2000 kilometers, same quality of the tomato, if you send it to 100 kilometers and 2000 kilometers, definitely the quality will be reducing, right. So we have to take the decisions. Biosensors will help us in taking the decisions, and shelf life prediction also, eco-friendly and non-destructive analysis. I told you biosensor, you whatever the parameters you are measuring using the biosensor, it is non-destructive process. These are some of the key components of the biosensor. The first one is bioreceptors. Bioreceptors, it includes enzymes, antibodies, nucleic acids, etc. Okay. So these are called as bioreceptors. Then after bioreceptors, we have a transducer element. Converts this biological response into an electrical signal. What will happen? Glucose, I am telling example, right? If glucose is converted as gluconic acid or gluconate, glucose is nothing but the salt of gluconic acid, it will take some process in time, right? Processing time. If you add enzyme in it, what will happen? Enzymes are catalysts, right? It will speed up the reaction. During this process, gluconate plus some byproducts will be released. H2O2 will be released, some redox mediators will be released, okay. So during that process, what will happen? Some electrons will be released, some protons will be released. So those things, if you identify, okay, definitely there is some electrical signal will be generated during that chemical process. That electrical signal we will be determining by using this sensors, transducers, right. The last one is signal processor, interprets and displays the data, okay. The bioreceptor is a molecule that specifically recognizes any analyte or substance, something like glucose or something like that, okay. So this is the overview. See, biosensors, in the left side you have bioreceptors. These are the bioreceptors. Using enzymes, okay, you can detect glucose or any physical, some nutritional component in the food sample. And then some electrochemical, piezoelectric, calorimetric, optical type of transducers are there. You have to interlink. You have to link these enzymes and the transducers to get the output, right? We will be learning all these things. See here, this is very good example. These are the analytes, right? Analytes means what? This is what you need to identify. In a food sample, take an example of juice. Juice has enzymes, juice has different components, okay. If you pass this juice analyte into the biorecognition element. Biorecognition element, bioreceptors can be anything. It can be enzyme, it can be antibody, or it can be microorganism, DNA, or any kind of cells. It acts like a lock and key mechanism, okay. Example, you take this key as an analyte and lock as a bioreceptor. You cannot insert any kind of key to unlock the lock, right? Specific key is required. So if you are measuring glucose, These bioreceptors will measure only specific glucose molecules only. These are highly specific in nature. It will eliminate all other molecules. Okay, that's how it works. Then the signal will be generated. Some electrical, charges, electrical current will be generated. That will be, given as a output. Okay, this will be detected by the transducers. Transducers, electrochemical. See, this is the detectors, transducers, and output. Okay, you will get the data at the end. Okay, see here we have analyte. Biochemical conversion is happening. Glucose to gluconic acid. Okay, using antibody bioreceptor or enzyme or any kind of bioreceptors, if you put it here as a thin sheet, so what will happen using these transducers? So if any kind of fluorescence is being generated or any color change is happening, using optical transducer, you can easily recognize, okay, this particular element is present. Okay, based on the output, based on the possibility of what reactions are happening, you have to decide these transducers. Okay, then after that you will generate the results accordingly. So these are uses of biosensor. The first one is quality monitoring, pathogen detection. In the food processing, this is very much important. Then nutrition assessment, real-time monitoring, and then non-destructive analysis. Okay, we'll go through different these bioreceptors. Okay, the first bioreceptor will be enzyme, and enzyme, using enzyme you can easily detect several components. Okay, so enzymes are biochemical, biological macromolecules with catalytic activity. I told you, highly specific in nature, responsible for speeding up the biochemical reactions, right? And then I gave you example. Okay, glucose oxidase. Glucose oxidase catalyzes the oxidation of the glucose, which produces the hydrogen peroxide. H2O2 will be produced. The decrease in the oxygen concentration is proportional to the glucose concentration. Okay, so the decrease in the oxygen concentration. So how much amount of oxygen is there? If the oxygen is reducing, or how much amount of hydrogen peroxide is generated. If you know the concentration of these two things, you can easily identify what is the glucose concentration. So other enzymes, something from horseshoe radish also we can extract some enzymes. From different agriculture commodity, different enzymes can be extracted. Okay, so using all these enzymes, we can identify several things. So how it got how this biosensor concept derived? The first, in 1962, Clark and Lyons, they invented this glucose biosensor. After that, the biosensor concept got emerged, and now it is talk of an or. Okay, everybody is talking about biosensors. Okay, so here the simple, if you see this glucose plus oxygen, okay, using this particular glucose oxidase enzyme, we are getting gluconic acid, glucose acid, or gluconic acid, or gluconate, all are same, plus H2O2. Using this simple reaction, you can design a biosensor. Okay, this is how it looked. See, oxygen, you must have seen, right? Glucose sensor, on top of the glucose sensor there are vents. Oxygen will be taken. Okay, this is that vent. From here, oxygen will be going inside. Electrode will be there. Electrode will give us how much concentration of H2O2 is produced. H2O2, see, near the electrode and then the oxygen, this particular layer, it has got some gap. Okay, so in here the electrochemical process will happen. Here the H2O2 will be generated. After that, it will go to the detector. So normally, see, earlier time 1962, platinum was used as an electrode. It was very costly. Now people are using graphite sheets, graphite electrodes. Okay, for cost reduction purpose. And then this is the working of glucose biosensor. Enzyme based reactions, enzyme gluconic acid to catalyze the oxidation of the glucose, and then oxygen dependency. This particular, it has a drawback. What is the drawback? The entire biosensor is depending on the oxygen. Oxygen acts as a natural electron acceptor in this process. Okay. Then detection of the hydrogen peroxide. Okay, the generated hydrogen peroxide is electrochemically detected at the electrode surface. That's what you have seen here. Here you will get the hydrogen peroxide. The current produced during this oxidation is proportional to the glucose concentration. How much amount of current is being produced? That is proportional to the glucose concentration in the sample. Okay. So this is the limitation. The performance of the biosensor depending on the oxygen availability and is affected by the interfering substances. Not only H2O2 will be produced. So this is very simple process, right? But in reality, this may not happen. Okay, this particular enzyme, it may react with some other molecules and it will produce H2O2, which has no relation with the glucose. So at that particular point of time, this particular glucose sensor, first generation sensor, will give the negative results. Okay, that is the limitation. To address that limitation, second generation sensors have been invented in early 80s. Okay, so here what is the thing? The limitation of the first generation glucose redox mediators carrying electrons from enzyme to the surface of the working electrode were introduced. So here redox mediators are introduced. What are these redox mediators? These are some of these components. Ferrocyanide. Okay, these are sometimes toxic, but in the second generation biosensors, people use these redox mediators to analyze how much amount of hydrogen peroxide is produced. Okay, and what is the benefit? Here, whatever the interfering substances, interfering elements, it will be eliminated. Okay, that is the major. This is how it works. Mediated electron transfer. Biosensors use mediators to transfer electrons from the enzymes directly to the electrodes. Okay, this is important. Enzyme directly to the electrode, bypassing the need for oxygen as an electron acceptor. In the first generator, without oxygen, we cannot able to produce the results. Here, bypassing the need for the oxygen as an electron acceptor. Okay, then enzymatic reaction will happen. All these remaining processes are same. Gluconic acid will be generated on the right side using the GOX, this glucose oxidase enzyme. Then electrochemical signals will be produced. Mediators is re-oxidized at the electrode surface, regenerating a measurable current. So what will happen? These these substances, these redox mediators, it will lose electrons, it will gain electrons. Okay, again how much it is losing and how much it is gaining. Okay, so that will be calculated and accordingly result will be produced in terms of numbers. Okay, next one we have third generation. Even that second generation, these are something like in mobile you have many applications, every time you will get updates. Similarly in the biosensors also updates are important, right? Whatever these redox mediators used in the biosensors, these are toxic in nature. Okay, if you discard that outside, okay, after maximum use of these biosensors, you will throw it outside, right? So it may pollute the soil or maybe cow or any kind of cattle. There is a chance of toxin, okay, toxication. So that's why in order to remove this highly toxic material, electron can perform direct electron transfer using this organic conducting material based on the charge transfer complexes. Okay, in the third generation, complete elimination of the redox mediators. First generation, there is no redox mediators. Okay, there will be interfering substances. The results will it may go wrong. Okay, it may not have any link with the glucose also. That in the second, using the redox mediators, interfering substances were eliminated. Even then, because of the toxicity, the third generation has been introduced. Okay, this is the nutshell. First, second and third, you will have electrode in each and every sensor, first, second and third. Here glucose to gluconate, some enzymes will be there. Oxygen, using oxygen you will produce hydrogen peroxide. Okay, here these are redox mediators. Okay, using this redox mediator directly, it is not depending on O2 and H2O2. Okay, directly the element will be here. Using this electrode you can easily analyze. In the third, there is no redox mediator, directly you can, using this particular system, third generation system, directly you can measure how much amount of glucose is there in our sample. So here measurement of current resulting from the redox reactions. Whatever the redox reactions are happening, it will produce some kind of electrons and protons. Because of that, current will be generated. Okay, it will trigger. Okay, then that particular current will be analyzed. Okay, so this is one example. This is glucose self-powered biosensor. Self-powered, there is no need of external any power, batteries, nothing is required. See here, the process is same. Glucose, it is mixing with oxygen, gluconic acid, gluconate, this is happening, it produces H2O2. Again this H2O2 is reduced to water, H2O. Okay, here this NAD and NADH, it produces some energy, right? In the ATP cycles we learned this one, right? NAD, NADH, okay, these things. And then glucose to gluconate here. Okay, this is the graphite sheet provided here. Again this is also one this electrode. Okay, between this, what is the potential difference happening? That is directly converted as the amount of glucose present in the sample. Okay, that's how these sensors work. Then what are the applications? Definitely you can use it for ethylene determination. Okay, monitoring the ripening and maturity. If you have this kind of cold storages, okay, banana or maybe any kind of fruits, this will release ethylene gases. Using these biosensors, you can easily detect the ethylene concentration. Enzyme based biosensor detect ethylene gases. Okay, what is ethylene gas? It's a ripening hormone using enzymes like ethylene oxide, oxidase or oxidoreductases. Using these two enzymes, you can easily identify ethylene detection. Okay, the next one is starch and sugar levels. Okay, enzymes like amylase and invertase, these can be used to measure the starch breakdown, okay, and sugar accumulation, indicators of fruit maturity. Okay, this is example I have given here. The next one is detecting the spoilage. So how do you detect the spoilage? Some during ripening or maybe during transportation, some of the volatile organic compounds will be released. Okay, volatile components like ethanol will be released. If it is fermenting more, or maybe bad fermentation is happening, or any kind of changes or acetaldehyde production, ammonia production, or any kind of microbial activity, it will produce some volatile substances. During using this biosensor, you can easily find out the volatile organic compounds. The next one is, see here, fresh apple and then spoiling apple. Okay, this is the spoiled apple. Germs, germs are attacking, started emitting the gases will be released. This particular gas will be identified using the biosensors. Enzyme activity of spoilage microbe, detection of the microbial, microbial enzymes, cellulases, pectinases. Why these enzymes are very much important? These enzymes are, cellulase, pectinase, these are plant, molecules, right? It will be present on the surface of the fruits. With time, what will happen? It will, release water. It will break down the cell wall matrices and release whatever water is present in the cell walls. Okay, that is not good. The texture, it will lose its texture, integrity will be lost. So that's why we need to, analyze what is happening with respect to the enzymes and microbial spoilage. The next one is nutritional quality assessment. Okay, in the nutritional quality assessment, vitamin C. Okay, these enzymes like ascorbate oxidase, it can be used for measuring the vitamin C. Then example is this one. So what is this? These are freshly cut or sliced or diced fruits or vegetables. With time, what will happen? With respect to time, see the color is changing, getting brown because of the enzymes, right? Which enzyme is responsible for dark, browning? Polyphenol oxidase, right? That is responsible. So how much polyphenol oxidase activity is happening? Using this biosensor, you can easily peroxidase based biosensors monitor oxidative reactions. Okay, and then which can lead to browning or loss of quality in stored peroxidase, polyphenol oxidase. These are some of the enzymes responsible for browning. Then next one is very much important, pesticide residue. Okay, so many pesticides are being used on the fruits and vegetables. Using these biosensors, you can easily detect how much amount of pesticides are there on the surface of the material. See, by naked eyes, we cannot tell how much amount of pesticides are sticking on the surface of the fruit, right? Some of the pesticides, these molecules, organic molecules, it will react with the the bioreceptors. Okay, once it is reacting, then some kind of conversion happen. That particular current, you can identify how much amount of current is being generated. Accordingly, you can get the numerical value. Next one is shelf life estimation. Shelf life estimation, respiration rate monitoring, and then polyphenol oxidase activity. All these things are very much important. Next one is disease and pathogen detection. Okay, so fungal and bacterial diseases, something like some bad bacteria is entering into the food system, that has to be identified. How that what is the substrate that particular bad bacteria is using in the food system. Okay, using this biosensor we can easily identify. Enzyme based biosensor detect lytic enzymes. Okay, chitinase or proteases or whatever different kinds of enzymes can be used. Then mycotoxin detection, mycotoxin, aflatoxin, these are common for nuts. Okay, sometimes what will happen, have you heard about mad cow disease? Okay, cow it eat infested aflatoxin contaminated corn cobs. If they are consuming a lot, then it will affect the brain of the cows. Okay, that's how these toxins, aflatoxins, are harmful even for our human body also, not only for the cattle. Okay, so detecting the mycotoxin, aflatoxin, whatever it's a broader term, mycotoxin. So how can you detect this one? Using these enzymes you can easily detect. Then it is also used for sorting and grading purposes. Okay, and then especially see here anthocyanin pigments. If you use any, so how do you measure the anthocyanin? Based on the pH differences, right? So we have different pH buffers will be there. So this is anthocyanin, right? This is what jamun fruit. Okay, using biosensors you can easily determine with respect to time how the color change is happening. That is also you can determine. Okay, then using this packaging, see here on top of the packaging, what you can do, you can coat this particular bioreceptors. When this packaging is coming in contact with some of the gases produced in the fruits, maybe suppose take an example, half kg of fruit you are packing in the packet, right? So with time what will happen, some gases will be produced. That gas, it will be reacted with the whatever the the sensors, enzymes or antibodies or whatever you are putting on top of the sheets, that will be reacted. Accordingly you can sense what is the shelf life of this particular fruit. These are some of the advantages. Okay, high sensitivity and high specific. Enzymes are highly sensitive and highly specific. If you are using glucose oxidase, it will only react with glucose, not other compound. That's why it is highly specific. You will get the rapid results definitely. Then it is a non-invasive detection, cost effective, integration with the IoT is also possible. Okay, these are some of the application. Apart from food industry, you can use these sensors in different fields. So in the food, you can see here contaminants, pesticides, antibodies, they are used for detection of ethylene, glucose, and glucose biosensor can be used for quality control in the fruit industries. Okay. The next one is antibodies as the bioreceptor, similar to the enzymes. These antibodies acts as a bioreceptors. Okay. So we will see how. See, suppose for example, these are analytes, right? It will come and contact. How this COVID is identified. COVID has this COVID detection, PCM of the technique. So it has some structure. Okay. So if you identify that particular structure, you can easily tell what kind of strain it is, either virus or bacteria or whatever it may be, right? These antibodies, it will have some structure. It will only identify specific structures. Okay, if this analyte has this particular structure, it will catch that particular analyte. It will not catch other thing. how many analytes are being trapped by these antibodies. You can easily quantify, right? Using this antibody based biosensor, you can easily identify how many analytes are in the system, food system. Then it will transducer element is common. Here only this will be changing, enzyme, antibody, any other cell, protein, whatever it may be. Transducer and data analysis is common. Okay. You can use DNA also. Sometimes DNA strands, RNA strands can be used as antibody, and then you can detect whatever the target element using this particular technique. Detection of pathogen, bacterial contamination is you can detect Salmonella, E. coli, Listeria monocytogenes. Okay, these are commonly prevalent in fresh produces, fruits and vegetables. Okay, then viral contaminants. You can also determine norovirus or hepatitis A that can contaminate the fruits and vegetables. Then this important is fungal spores. Okay, some of the fungal spores you can easily identify to reduce the post-harvest diseases. Okay, and then monitoring the mycotoxins. Here enzymes can also be used, and this also can be used. Then pesticide residues. Sometimes farmers, they use a lot of pesticides that will be coming in contact with the fruits or vegetables that you are harvesting. It will be sticking on the surface of the commodity. If you are not washing, even if you wash properly, some traces will be sticking on the surface. If you are eating, it will definitely show adverse effects. Just like antibodies, analytes will be coming and sitting with the antibodies, just like lock and key mechanism. This harmful substance will go into our body. It will be opening the cells and entering into our system. Okay, that's how it happens. Then we'll see these symptoms. Okay, whatever vomiting, nausea, whatever the symptoms you get. Okay, after toxication, all these things are possible. Then quality and freshness monitoring. Ethylene detection. This particular sensor can be used for ethylene detection, spoilage indicators, enzymatic markers. Okay, enzymatic markers is also important. Some of the application in food related application, this is important. Salmonella identification, E. coli, Listeria, identification of allergens, peanut, gluten. Allergen identification also we can use this particular biosensor. Okay, suppose few of us sitting here are lactose, allergic to lactose or allergic to gluten. Okay, allergic to some different components. We can easily identify what kind of allergens are available in the particular food system. Okay. These are some of the advantages. These will be similar with these enzymes. Okay, highly specific, definitely. This is also highly specific, just like enzyme. Antibodies also highly specific, rapid and real-time results can be generated, non-destructive, portable and user-friendly, compliance with the safety standards. Okay, this is also very much important, right? Safety standards are very much important. Help ensure produce meets regulatory limits for contaminants and allergens. Okay. Why the safety standards are important? FSSAI, they have some guidelines. Okay, or maybe this medical related guidelines will be there. If you are using this particular biosensor, you have to go through all these protocols, and accordingly you need to use your this one biosensors. Okay. Next is nucleic acid. Similar to previous one, nucleic acid can also be used as a biosensor. This is how it works. Okay, this peptide nucleic acid bioreceptor, if suppose for example strain DNA is there, if you want to identify some different nucleic acid, it should have the compatibility, complementary DNA, we call it as complementary DNA. DNA or RNA hybridization is the process of complementary DNA and RNA strands bonding together to a double stranded. If your target is matching with whatever the bioreceptors you have, okay, just like enzyme you can also have DNA strand, but it will be single strand. The component that you are identifying, it will be coming and it will be complementary with this DNA that you are giving. Okay, then it will have a perfect strand, fulfilled DNA strand. Accordingly, the voltage will be generated. You can measure the current will be generated. Accordingly, you will get a graph how much amount of the particular DNA RNA hybridization happened. You can easily identify. These are some of the applications. You can see detection of again this foodborne pathogen. Here the genetically modified organisms, something like the genes of brinjal, GT brinjal, or any kind of genetically modified plants are there. You can easily identify those by using this particular sensor, nucleic acid sensor. Many advantages are there: specific, versatility, sensitivity, and then you can customize this particular nucleic acid sensor. Okay, you can just change the DNA strand, and then accordingly it will react. Labeling flexibility will be there, small in size. Okay, these are some of the advantages for this nucleic acid. Okay, these are all about the the whatever related to enzymes, nucleic acid, bioreceptors. Once this bioreceptors in contact with the target component substrate, it will generate something, right? Electricity will be generated, maybe some illumination will happen, some changes will happen. Those changes can be identified using these transducers. Okay, there are 4 different types of transducers are there. We will discuss one by one. Electrochemical transducers, optical, piezoelectric, biosensors. Okay, see here electrochemical biosensor detects the electrical parameters related to chemical reactions. Okay, when there is a chemical reaction, some electrical parameters will happen. Those things you can identify. Okay, analyzing the electricity as an outcome of the chemical process. Okay, this is important. Then electrochemical biosensor is based on the electrochemical transducers, providing selective and quantitative analytical information. Then this particular bioreceptor serves as a recognition element that binds or transforms the analyte. This is very important. It binds the element or transforms the element according to the requirement, while the electrochemical transducer converts the event of bioreceptor analyte interaction into an electrical signal. Right. So this is what happened. Sample, we have sample, biorecognition element, transducers. Here by using change in the current, change in the voltage, those things we can easily identify. Okay, then there are again in the electrochemical techniques we have different using potential voltage change, potentiometry and voltmetry, impedance and ampermetric. These are different by using current you can identify, using voltage you can identify, change in the voltages, differential voltages you can identify. There are different methods are there. It got evolved with respect to time. See chemical, how it works. Chemical reactions will happen. Target molecule analyte interacts with the biological sensing element. Okay, then electron or ion generation. The reduction produces specific byproducts, just like in the glucose, this one hydrogen peroxide produced, right? So here specific byproducts will be produced, such as ions or electrons, or molecules like H2O2 will be produced, that you have to identify. Then transducers detect these byproducts and convert their activity into electrical signals using this particular techniques, using the ampermetric potential, using the resistance also. Okay, you can resistance is nothing but impedometric technique. Different instruments will be there for identifying all these things. Okay, then the signal proportionality. In the signal proportionality, see more glucose means higher H2O2 production. How much H2O2 is produced, accordingly you can correlate, right? So that is signal proportionality. The next one is data analysis. You use different data software, how much current is being generated, and then accordingly you will try to correlate. Okay, what are the meaningful concentrations or values for the analysis. Those things we can easily identify, predict. Okay, then these are the things I told you, right? In potentiometry, potential of the electrochemical cell under static conditions is measured, whereas in the ampermetric the potential of the electrode is held constant. The slight differences are there. See, impedance means used electrochemical impedance spectra, EIS. This is commonly used for this particular technique, impedance technique. The relationship between the alternative current and the applied sinusoidal potential in a frequency domain is measured. Okay, the next one is voltmetry. Differential potential voltmetry, different voltmetry techniques are there. The current change is analyzed under the control, but not constant potential. Okay, the potential changes will be there. Those things we can measure using this electrochemical of optical biosensors. Okay, optical biosensors are majorly used for determining this water pollutants. Spoilage detection, ripening monitoring, nutritional quality assessment, pathogen detection and environmental monitoring. In the environmental monitoring, see, monitoring storage conditions, humidity, light intensity to ensure the optimal post-harvest quality is very much important. These are some of the classifications of this optical. There are so many, different updates have come. Okay, all these things are based on those updates. Okay, you can see, based on the fluorescence, based on the, the surface plasmon, and then absorbance, reflectance based, Raman spectroscopic based biosensors are there. Okay, and what we need to, do here is we need to identify how much cost effective these particular things are. Based on our usage, we need to see cost effectiveness also, right? That is also very much important. Accordingly, you can select whichever you want. The next one is piezoelectric biosensors. These are, analytical devices that utilizes the piezoelectric effect, where mechanical stress generates an electrical signal to detect the biological chemical components. These are also highly specific, and this is the working principle, how it works. It measures the mass change. See here, you can see here, it detects the mass change. How it detects the mass change? We have a piezoelectric material here. On top of the piezoelectric material, some antibodies or enzymes or some this bioreceptors will be there. You can design this particular thing. Accordingly, the target antigen will come and sit on this. Okay, that according to that, see here the frequency will be generated based on the mass. How many analytes, target antigens are sitting here? Based on their mass, we can easily identify. Corresponding voltage will be generated. Okay, see here mass loading, when an analyte binds to the sensor surface. Okay, sensor surface, this may be quartz crystal also. This is piezoelectric material, right? Quartz crystal is piezoelectric. It increases the mass, causing changes in the oscillation frequency and acoustic wave properties. Okay, this is how this particular system works. These are some of the applications. More or less, the application remains the same. Okay, these are classifications. These are just like our previous biosensor. We have different classifications where based on the quartz crystal, surface acoustic wave biosensor, different types of biosensors are available. These are some of the advantages. What are the challenges and limitations? This is very much important. We have seen, so far bioreceptors we have seen, and then different transducers. Okay, how the transducer works. Okay, based on the changes, the electrochemical this one process, chemical changes will happen. So, what are the challenges and limitations of this? Calibration and standardization issues will be there definitely. Okay, glucose sensor. How many times you can use the glucose sensor? Single time. That strip has to be changed. Am I correct? You take out the blood sample, you put it on the strip. You are sending that strip into the transducer. Accordingly, you will get the value. Okay. Similarly, calibration also very much important. Whether that value that you are getting it is calibrated or not. Sometimes in the watches, we have the smart watches, right? It will give us blood pressure. Okay, whether that is correct or not. If you test in the hospital, your blood pressure is different and you are getting different blood pressure in the watch. That is also possible. That's why calibration is very much important. Limited shelf life of the bioreceptor, high initial cost of development, then potential for false cost or negative result. This is very much important. Whether we are getting the right positive or right negative results, that we need to analyze. Need for skilled operators. Everything is involved directly or indirectly with the cost. Okay, if you are planning to use biosensors in some industry, definitely costly process. What are the recent innovations? So, nanotechnology in the biosensor is the recent innovation. Okay, then development of wireless biosensors for remote monitoring. Okay, wherever you are sitting, you can monitor your glucose levels or maybe whatever levels. Okay, in the sample. Integration of Internet of Things for real-time data, miniaturization. Okay, and wearable biosensor. Wearable biosensor watches. All these things are wearable biosensor. You can keep that biosensor in the ethylene ripening chambers or maybe any kind of storage chambers like that. Multi-analyte detection in a single device. Okay. These are some of the recent future scope. Commercialization of low cost biosensors for small scale farmers. This is required because biosensors everything is interlinked with cost. So you can plan for this expansion to detect the broader range of analytes. Then integration to smart packaging system for freshness monitoring. You must have heard RFID tags. You can adopt biosensors. If the color is changing, you can identify whether this is ripened or matured or what has happened. Okay, this is one example I am telling. Using different types of biosensors, you can easily predict what is happening with the food, right? Then advances in AI and machine learning for automatic, decision making, role in sustainable food systems and reducing global waste. All these things are some of the future scope. In nutshell, if I tell biosensors, biological recognition elements, physical transducers. Okay, enzyme based are there. These things we discussed, enzyme based, and then cell based analytic, this DNA strands, all these things we discussed. Then antibodies, reserved nucleic acids we discussed. Then in the transducers, this electrochemical transducers, optical transducers. This is very vast topic. Okay, in one class if I tell this is what I can do. This is the maximum justice I can do for this. Then piezoelectric and thermal sensors. Okay, biosensors are transformative tools in post-harvest management. They ensure food safety, quality, and sustainability. This is, in one line, if I want to convey you people what is biosensor, these two things. These are definitely transformative tools in the post-harvest management. Okay. With this, I would like to conclude my presentation.